Good morning, friends. Today we are going to learn the gene concept. That means we need to understand the definitions of gene, genome, chromosome, what's the modern concept, how the concept of gene has been evolved historically, and most importantly, what are the major differences between prokaryotes and the eukaryotic gene structure. So, all these are covered under the subtopic of unit number 3 of cytogenetics. Okay, so we are going to start the first one, genome. We are going to start the genome. What is the genome? Genome means a set of all the DNA found in an organism or its all component cells. That means what? That means that genome is the hereditary basis of every living organism. Okay, so consider the case of prokaryotes where the genome include main chromosome and the different kind of plasmid, because all those are basically nothing but the DNA. Similarly, in case of eukaryotes, we know very well that in case of the animals, the DNA is found in nucleus as well as in mitochondria. But in case of plants, it is found in mitochondria, nucleus, chloroplast, etc. In case of yeast, there is also a kind of plasmid. So all this set of DNA, what is the meaning of the set of DNA? That means, for example, human beings are diploid creatures. So there are the two set. Okay, we are just talking about one set. That means the haploid number, haploid chromosomal number, haploid DNA molecule number are collectively called genome of an organism. Okay, so in this diagram you can see here, say this is the whole genome, and that part of the genome is a chromosome, and in this chromosome there are many genes. Say this particular part is a gene. So gene is nothing but it is a part of DNA sequence of chromosome, and then again there would be the certain information within the gene that where should the transcription or the expression of a gene should start or where should it end. So whole the procedure, whole the information are basically contained within the gene. So moving to the next one, we are going to consider how the gene concept has been evolved. See, since the Mendel's time, it was called factor. After that, it was called gene. So the point was, simply Mendel knew and Mendel proposed directly or indirectly that a gene or a factor is nothing but a genetic level something factor kind of situation that control a character, of phenotype. Phenotype means the visible situation, the appearance. Okay, so firstly, while working on the alkaptonuria, the Beadle and Totem firstly proposed one gene, one enzyme concept through their mutational studies. What they did? They simply did mutations in an enzyme and they found that the metabolism is hampered because that enzyme is not working. So it was a straightforward correlation by them that a gene must encode an enzyme. But thereafter, further studies have improvised the definition, and many of the scientists thought that there are thousands of the proteins which are actually heteromultimeric. Heteromultimeric means they are composed of more than one type of subunits, and each type of subunit is encoded by a gene because it is having a discrete information. So, for example, in case of globin gene, globin protein, that is a part of hemoglobin, there are 4 polypeptides, two alpha polypeptides and two beta polypeptides. And since these are the two different polypeptides, that means they are having two different discrete information. So they must be encoded by the separate genes. Okay. So this concept, one gene, one enzyme concept has been further improvised into one gene, one polypeptide relationship. But still there were questions that there are many genes which it doesn't necessary for them to encode polypeptide only. They may encode the functional RNA molecule. So they are ultimately not translated into polypeptides. For example, we know very well rRNA. We know very well about the tRNA. They are having a kind of information and that is very important for the protein synthesis. But they themselves are not translated into protein synthesis. So it is not necessary that a gene must encode a protein. Rather than we should conceptualize this one as a gene is nothing but a genetic information that specify a particular function. So considering that aspect, but before going into that one, we can understand or we should know that how the gene concept has been evolved. 1903 chromosomes are hereditary units, it has been established. See, it is around 110 years ago information, and still we are on the work in molecular biology. In 1910, it was found that genes lie on chromosome. That means the genes are the part of chromosome. It has been known. In 1945, gene codes for protein. That means one gene, one enzyme hypothesis, Beadle and Tatum hypothesis, has been proposed around 1945. That means in 1940s. Now, in 1977, eukaryotic genes are interrupted. That means a split gene concept has come into light. And finally, genome sequenced for the prokaryotes, it was in 1995, and for the human, it was 2001. So it was a historical account, nothing important as such. Now moving to the modern gene concept. Modern gene concept, as we have already discussed, a gene encodes a RNA, that we know very well because no DNA sequence is directly converted into proteins. So there must be a mediator kind of situation, and that mediator is nothing but the RNA. And especially this is the messenger RNA, mRNA, which in turn encode a polypeptide. Most of the genes are structural genes that encode polypeptides, but some encode RNAs. And the role of the RNA is not the structural one. These RNA are different from this RNA. This was carrying the message to encode the polypeptide, while these RNA which are not translated into polypeptide, rather than these are having the regulatory role. For example, miRNA, the microRNA, siRNA, short interfering RNA, gRNA, guide RNA, that is responsible for the RNA editing. Another one, tRNA, transfer RNA, rRNA, ribosomal RNA. So considering these two, tRNA and rRNA are having the role in the protein synthesis machinery. And these three, at least three regulatory genes. With the time of research, we still got the information about another kind of RNA which are having the regulatory role. For example, long RNA like this, short interfering RNA, long interfering RNA have been discussed. Okay, there are another kind of RNA, for example, snRNA, snORNA, which are having the role in the RNA processing. So there are 1,000 kind of RNA. Those many of have been discovered and many are still in the pipeline. So ultimately what happens, we can simply summarize by this diagram. Gene is nothing but a DNA tract which are having the mainly two parts. One is the structural gene, which is basically transcribed into an mRNA. Okay, and the another one is the regulatory part of the gene, that is usually called the promoter. From here this transcript has been started. So from here this side is called downstream side and this side is called upstream side. You can consider this particular part of the DNA, this one from -35 to this arrow. You can consider this is the transcription unit because it is having the information of termination of transcription, start of the transcription. This is the coding part and that is the regulatory part called as promoter. So what's the ultimate definition comes our mind while considering all these modern aspects and the research? We can say that the gene is a DNA sequence that specifies an independent functional product that may be polypeptide or RNA, whatever the kind of RNA. That means this is functional, this is functional. That means gene is nothing but the DNA sequence which specifies a functional product, whether in terms of RNA, any kind of RNA, or in terms of polypeptide. So that was the overall modern concept of gene we can consider. Next part we are going to consider, that is the eukaryotic gene structure. In case of eukaryotes, what happens? That the coding part, that means the DNA sequence that specifies amino acids, that is found interrupted by non-coding part within the gene. And these things have been, these intervening sequences that have been mentioned here are called introns. As opposed, the coding part which is going to specify the amino acid, these are called exons, that means expressed sequence. So more than 75% we can consider eukaryotic genes are having the interruption in their coding region because of the presence of introns. That's why eukaryotic genes are also called split genes. That means their information for the coding of a protein has been splitted. Okay. Many are the proteins which are very large in size. For example, we are going to consider titin. Titin is a protein found in the muscles, human muscles, and that is the largest protein found reported till date in human beings. Its gene has approximately 233, that means 233 introns, and plus one are the exons, that means 234 exons are there. So considering the another gene, for example globin gene, globin gene is containing 2 introns. So what we can conclude? We can conclude that eukaryotic gene may have multiple exons or introns. It totally depends on a type of gene. And sometimes it also been found interesting that the combined length of the introns are found much greater than total of exons. That means what? These 2 introns, if are found present, it may be the possibility that their combined length is greater than that of the combined length of 3 exons. So this is the most important aspect we can consider. Such kind of introns have not been reported or rarely reported in case of prokaryotic genes. That means these are more or less exclusive features of the eukaryotic system. But in case of eukaryotes, we should point out here that mitochondria and chloroplast have their ancestry among the prokaryotic setup, and that's why they are carrying the circular plasmid and circular DNA as well. That circular DNA is not having introns because it is of bacterial ancestry. So these introns have been reported in case of what? Mainly nuclear genes. But with the recent reports, it has been noticed that other kind of introns have been reported in mitochondria, chloroplast, some of the bacteria, and these have been classified as group 1 and group 2 that we will discuss in the next chapter regarding the introns. So we have noticed that the introns have been the interruptions in the coding region. So what is the requirement? The requirement is that must be removed while specifying a protein. So there are the two possibilities of the removal. Within the gene, they can be removed, or their transcript has been designed and from the transcript they have been removed. So this pathway has been selected by the nature. That firstly, the whole exon intron sequence is transcribed into pre-mRNA, and that pre-mRNA is finally spliced and introns have been removed. The spliced mRNA is the functional mRNA. That means this is not functional. This is only the functional. That means intronic sequence part must be removed before its translation. So we have discussed about the introns and why, what's the significance, that we will deal in the next chapter. So thank you so much. Stay blessed, stay happy.